Hello, we'll start now. Hello. Hello.
Yes, ma'am. Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Unit 1, your unit 1 is cell and cell organelles. So you can see the screen, right? The screen, can you see?
Yes, ma'am.
Okay. So in the cells and biomolecules, we will learn what is the main domain of life and basic structural unit of all living organisms and their biochemical composition of the cells, and differentiate between a plant and animal cell. And so these are the things we will cover in this unit 1. As you know, the cell is the basic unit of all the life forms present in our earth. So composition of cell was first used by Robert Hooke in 1664, and now we will know the biochemical composition of the cell. What is the biochemical composition? Why the elements present in the cell are oxygen, carbon, hydrogen, nitrogen, calcium, phosphorus, and the table. This table has been given here. This is the table. The elements oxygen, carbon, hydrogen, nitrogen, calcium, phosphorus. This is the chemical composition of the cell, and there how much oxygen percentage, how much carbon percentage, hydrogen, nitrogen, calcium, and phosphorus percentage in a cell. It is given here. And sulfur, potassium, sodium, chlorine, magnesium, iron, zinc, copper, manganese, etc. How much percent by weight, it is given here. And water constituents nearly 60 to 80% of the weight of the living organism. Then what is domains of the life? The earlier biologists believed that all life on the earth belong to one or two primary lineage in the eukaryotes, which include animals, plants, fungi, and some single cell organism is called prokaryotes, which include bacteria and all living microscopic organisms. And these prokaryotes actually have the two different groups, eubacteria and archaebacteria. There are the two bacteria present in the prokaryotes, and the newly recognized latter named archaea. Archaea are aquatic or terrestrial microorganisms that differ both biochemically and genetically from the true bacteria. And archaea bacteria and eukarya, there are two classification of trees of life consist of three domain: archaea, bacteria, and eukarya. These archaea and bacteria are prokaryotic single cell organisms whose cells have no nucleus. It's archaea bacteria, it has the no nucleus. Cells of the eukarya possess a nucleus and membrane bound organelles. The animals, plants, fungi, and protozoa are included in the eukarya. Based on the degree of complexity, the cell organization, the cells can be classified as prokaryotes and eukaryotes. Prokaryotes, some basic common properties, some of which are given. All the cells store information in the genes. Ribosome synthesis protein in all cells. Protein control the structure and function of the cells. ATP is the molecule used by all cells to transfer energy. Plasma membrane is the common feature in all the cell. All the cell plasma membrane, in every cell plasma membrane is there, which is a common feature of the cell. A protein is there, which control the structure or function of the cell. Structure of the prokaryotic cell. Structure of the gram-positive bacteria give distinctive purple appearance, having gram staining, and gram-negative bacteria give pale reddish color after gram staining. Prokaryotic cell are single-celled microorganisms known to be the earliest living organism on the earth. Prokaryotes include bacteria and archaea. Then what is prokaryotic cell? Prokaryote include bacteria and archaea. The characteristic of the prokaryotic cell are that they lack nuclear membrane. Prokaryotic cell, they lack nuclear, there is no nuclear membrane, and membrane bound organelles such as mitochondria, Golgi bodies, chloroplast, and lysosomes. The DNA present in the region in the cytoplasm without a membrane and is known as the nucleoid. Then the cell, what is cell wall? Cell wall, it is made a thick layer of the protein and sugar that prevent the cell from the bursting and made of rigid framework is called the peptidoglycan. What is plasma membrane? Plasma membrane is called the cell membrane, is present at the inner surface lining of the cell wall. Nucleate. Within the cytoplasm lies a single molecule of the double-strand DNA in the form of an endless loop is called the circular DNA. And structure of the eukaryotic cell. Structure of the eukaryote have a clearly defined nucleus. Eukaryotic cell also contains organelles including eukaryote has mitochondria, endoplasmic reticulum, and Golgi apparatus and lysosomes. Like digestive apparatus within many cell types. Composition of the cellular organization, prokaryotes, it has cell size 1 to 10 micron in diameter, metabolism, anaerobic or aerobic, organelles, few or non-organelles are there. Nucleus is absent in prokaryotes, nucleus is absent, eukaryote, nucleus is present. In prokaryote, cell division binary fission, and it is eukaryotes, it is mitosis or meiosis. In the there is difference between prokaryotes and eukaryotes. Prokaryotes, mitochondria absent. In eukaryotes, mitochondria is present. In the prokaryotes, cytoplast is absent, and eukaryotes, it is present. Cytoplasm is present in plant cell only. In the prokaryotes, cytoplasm has no cytoskeleton, and eukaryotes, well-defined cytoskeleton protein filament is present. And the cell organelles and their functions. In all the cells, whether prokaryotic or eukaryotic, all are bound by a limiting membrane called cell membrane or plasma membrane. The cell membrane gives shape and mechanical strength to the cell and protect it from the environment. In plant, the cell membrane is covered by a rigid cell wall in which the major component in the cellulose. Plant cell membrane covered by a rigid cell wall, which is the major component, it is called cellulose. In fungi, the cell wall is made of chitin. Difference between animal cell and plant cell. The cell membrane is composed of lipids and proteins, and in many cases 2 to 5% carbohydrates are also present. The carbohydrates are linked to the protein or lipids. Among the various models proposed for the plasma membrane, the most satisfactory model that is accepted is the fluid mosaic model of S.J. Singer and G.L. Nicholson. What is nucleus? In all eukaryotes, nucleus presents is the most prominent and conspicuous organelles of the cell. The nucleus being the control center of the cell. Nucleus is the control center of the cell, contains chromatin. DNA associated with a set of 5 basic protein is called the histones. DNA, what is DNA? DNA is the genetic material that is passed on from one generation to another. A DNA molecule with protein bound to it is known as the chromosome. The nucleus is filled up with a fluid is called nucleoplasma, which contains a roughly spherical body, the nucleus within which the RNA component of the ribosomes is synthesized. What is endoplasmic reticulum? The cytoplasm of most eukaryotic cell contain a very complex network of internal membranes called the endoplasmic reticulum, which form a channels and vesicles within the cell. There are two types of endoplasmic reticulum. What are the two types of endoplasmic reticulum? Rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is named for its rough appearance, which is due to the ribosomes attached to it. These are actively involved in the synthesis of protein. Protein that are meant to be exported out of the cell or that become part of the membrane. The smooth endoplasmic reticulum involves synthesis of lipids, including cholesterol and phospholipids, which are used in the production of the new cellular membrane. What is Golgi bodies? The Golgi bodies appear within the cell as stacks of flattened vesicles. Many of the protein synthesized by the cell have to be secreted out, as in the case of the hormones. Golgi bodies carry out this process by introducing certain specific and different chemical signals, carbohydrate residues on the proteins in a process called glycosylation. Glycosylated proteins are recognized at a different sites within the cell. Golgi bodies play a key role in chemically modifying newly synthesized proteins. Proteins secreted from the Golgi apparatus are directed to lysosomes or to the cell membrane. Still others are destined for secretion to the cell exterior. What is lysosome? The lysosome are present within specified membrane bound vesicle is called lysosome. Some 40 enzymes are known to be present within the lysosomes. Lysosomes are actually formed by budding from Golgi bodies. What is peroxisomes and glyoxysomes? A number of metabolic reactions such as oxidation of amino acids and lipids results in the production of the hydrogen peroxide. The hydrogen peroxide is extremely toxic to the cell, and oxidative reactions are confined within the cell to small membrane bound organelles called peroxisomes. In plants, the corresponding organelles are called glyoxysomes. What is cytoskeleton? All the eukaryotic cells have distinct shapes and are also capable of assuming different shapes. The internal organelles of a cell are also capable of migrating from one location to another manner. The cell is able to achieve these tasks by an internal scaffolding system is called cytoskeleton. And Then mitochondria, the eukaryotic cells, living organisms, continuously carry out a huge number of chemical reactions to live, grow, reproduce, and fight off diseases. All these processes require energy at the cellular level. Each cell that engages in any of these activities gets its energy from the mitochondria, tiny organelles that act as the cell's powerhouses. Mitochondria is the cell's powerhouse. An important function of the mitochondria is derive energy from different oxidizable nutrients such as carbohydrates, fats supplied to it. The mitochondria is able to perform this function by carrying out oxidation of these nutrients, giving metabolism, and energy released during such oxidation is transferred and stored in compounds called energy-rich compounds. Mitochondria are the largest organelles in a cell with a diameter 0.5 to 1 micrometer, and their numbers vary from one cell type to another. Mitochondria are the different shapes, sphericals in the liver cells, cylindrical in the kidney cells, or thread-like in the fibroblasts. Then what is chloroplast? One of the most distinguishing feature of all the eukaryotic plant cell is that in addition to mitochondria, they contain special light harvesting organelles known as chloroplast. These are the largest organelles in the plant cell. Chloroplast is the largest organelles in the plant cell and are about to 5 to 8 micrometer long and 2 to 3 micrometer thick. Chloroplast figure is there. And chloroplast about 50%. This is the representation of the chloroplast, and 50% of the dry weight of the chloroplast is protein and the rest is mostly lipid. The lipid material of the chloroplast consists of chlorophyll, carotenoid, and phospholipids. Chloroplast, like mitochondria, possess a bilayer membrane. So difference between the animal and plant cell. What are the difference between the animal plant cell? Cell wall? Animal cell wall is absent. Plant cell it is present. Plant cell wall is called cellulose. In animal cells, cell membrane is present. Plant cell it is also present. Flagella may be absent in the animal cell, absent in the plant cell also. Microtubules present in the animal cell, present in the plant cell. Golgi bodies present in both animal and plant cell. Centrioles present in the animal cell but absent in the plant cell. Nucleus is present in the animal cell, present in both the animal and plant. Both the cell nucleus is present. Mitochondria, both both animal and plant cell, mitochondria is present. Chloroplast is absent in the animal cell, but chloroplast is present in the plant cell. Chromosome present in both animal and plant cell. Ribosome is present in animal cell, present in plant cell also. Lysosome is also present in the animal cell and plant cell. And so these are the difference between the animal and plant cell. What is present in the animal cell? The table is there in the page number 24. The table is there. Difference between what is the difference between animal cell and plant cell? So in this unit, actually you learn the cell is the fundamental structure and functional unit of an organism. Cell is the fundamental structure and functional unit of an organism. Lipids, proteins, nucleic acid, carbohydrate constitute the four major classes of the carbon compounds found in all organisms. Then prokaryotes possess within the cytoplasm a single cellular molecular DNA not bound by the nuclear membrane. The various organelles in a eukaryote cell have their own identity and perform distinct functions. The ribosomes associated with the endoplasmic reticulum are the chief sites of the protein synthesis in a cell. Golgi bodies are the key elements involved in the processing and sorting a newly synthesized proteins. Microtubules, intermediate filaments, and microfilament, the structural element of the cell cytoskeleton, provide rigid framework to the cell. The plant and animal cell differ in some respects from each other, that is, in exterior structure, interior structure, and the cell organelles. For example, the cell wall in the plant cell is unique. Similarly, chloroplast and the vacuoles are absent in the animal cell. The various organelle of a cell can be fractionated by differential centrifugation method, which is based on the difference in the density of the organelles. So this is the unit 1, which cover the function, fundamental structure and function of the cell. And unit 2 is carbohydrate and lipids. Carbohydrate, what is carbohydrates? Carbohydrate are the form of most abundant class of the organic compounds. Carbohydrates are the most abundant class of organic compound. They are the compounds containing carbon, hydrogen, oxygen. Hydrogen, oxygen in these compounds are generally present in ratio 2 to 1 atom as in water. There are carbohydrates can be divided into classes based on the size of the molecule. Monosaccharide. The basic unit of carbohydrate has formula CN, HCN, H2N, ON, where N can vary from 3 to 8. Depending on the number of carbon atom, they are called as trioses. Trioses means carbon number 3. Tetroses, where carbon, there are 4 number of carbon atom. Pentoses, 5 number of carbon atom. Hexoses, 6 number of carbon atoms, and so on. The disaccharide consists of two monosaccharide units. When the two monosaccharide join together, it is called disaccharide. The oligosaccharides include the sugar up to 10 monosaccharide. When 10 monosaccharide molecule joined each other, it is called oligosaccharide. And polysaccharide made up of monosaccharide joined together but may contain thousands of units. Thousands of the monosaccharide when they join, it is called polysaccharide. These are the structure of these are the some monosaccharide: aldose, ketose, aldotriose, aldoses, aldopentoses, aldotetroses, aldohexoses. These are some monosaccharide. There are when thousands of these type of monosaccharide joined, it is called polysaccharide. Oligosaccharide, when 10 molecules of the monosaccharide joined, it is called oligosaccharide. And when only two molecules of monosaccharide join, it is called disaccharides. Then what is protein? Proteins are complex, diverse and high molecular weight polymers found in the cell. So in every cell, the proteins are there which are high molecular weight and polymers. The monomeric unit of proteins are amino acid. The monomeric unit of protein is amino acid. Amino acid joined each other in the specific manner to form an unbranched linear polymer known as the polypeptide. Thousands of proteins are present in a body are made up of only 20 different naturally occurring amino acids. Then what is nucleic acid? Nucleic acid are large complex polymer like protein. There are two types of nucleic acids: ribonucleic acid RNA and deoxyribonucleic acid DNA found within the cell. They are made up of four type of nucleotide. Each nucleotide is made up of sugar, phosphate, and a nitrogenous base. There are only two kind of sugars in the nucleotides, ribose and 2-deoxyribose. There are five types nitrogenous bases: cytosine, thiamine, uracil, adenine, and guanine. There are four nucleic acids: nitrogenous base, cytosine, thiamine, uracil, adenine, guanine. The nucleotide are joined together by a specific link 5,3 phosphodiester linkage to form linear on-branch chain of the polynucleotide. Then what is lipids? Lipids are heterogeneous group of compounds usually formed by the combination of a fatty acid with a glycerol. Fatty acids are the characteristic component of lipids present in both animal and vegetable fats such as ghee, coconut oil, and mustard oil. Lipids. Fatty acids are long linear chain organic compounds containing 4 to 24 carbon atoms. Uh, Nearly all the fatty acids in nature have an even number of carbon atoms, but those with 16 and 18 carbon atoms are most abundant oil. Fatty acids have a single carboxyl group and a long non-polar hydrocarbon chain. The presence of non-polar chain makes lipids insoluble in polar solvents such as water. And the water living system. What is ionization of the water? When water dissociate, it gives H plus ion and OH minus ion. H plus, OH minus. You see ionization of water. When water dissociate, it gives H plus and OH minus. The equilibrium constant between the dissociated molecule may be represented as H plus the OH minus divided by water. This is the equilibrium dissociation constant. K equivalent equal to, it is called equilibrium dissociation constant. H plus, OH minus and water in the bracket indicate molar concentration. This bracket is called molar concentration of the H plus ion, molar concentration of the OH minus ion, and molar concentration of the water. This can be written as water concentration, it is the molar concentration 55.5 into K equilibrium dissociation constant equal to H plus OH minus concentration, or this is called Kw. Kw is the ionic product of water. Kw is equal to H plus and OH minus. Kw is estimated as 1.0 into 10 to the minus 14 at 25 degrees centigrade. Since the concentration of both H plus OH minus is exactly equal, H plus equal to OH minus concentration equal to 1 into 10 to the power minus 7 moles per liter. Taking logarithm, it equal to minus log Kw equal to minus log H plus is equal to minus log OH minus H plus OH minus equal to 14. You know that all the biochemical reaction takes place at a specific pH. The pH scale is based on the ionic product of the water. You are familiar with this concept, but we recall for the benefit of all. Hydrogen ion concentration. pH is equal to hydrogen ion concentration. The acidity or basicity of a solution can be calculated in terms of the hydrogen ion concentration. Concentration of the hydrogen in a solution generally represented in terms of the pH. pH is defined as the negative logarithm of the hydrogen concentration. pH is equal to minus log H plus is the negative logarithm of the hydrogen ion concentration. This bracket is called concentration is equal to minus log S plus ion concentration is equal to 10 to the power minus 7 equal to 7.0. It is important to note that pH may vary only between 0 and 14 and 7.0 is the neutral pH value. Neutral pH value we called it 7.0. The pH scaling along with the pH, if it is less than 7 it is called the, this is acidic, and it is more than 7 it is basic. This is acidity. You can see the acids, it is These are the examples. This is neutral 7 at the point 7. It is neutral. Pure neutral is pure water 10 to the power minus 7. And this is bases. Bases is sea water, baking soda, alkaline desert pond, liquid soap, and acids are 1 mcl, gastric juice, lemon juice, vinegar, tomato juice, black coffee, rainwater. These are acids. Okay. Then what is buffer solution? Buffer solution is the one which changes in the pH upon addition of small amount of strong acid and strong base. The function of a buffer solution is to maintain effectively the pH of a solution. By neutralizing the acid or base added to it, a buffer solution is made up of two components, one of which is free acid or a weak base and other is salt. Two types of buffer solutions are generally prepared. Buffer solution type 1 buffer solution or acidic buffer and basic buffer. It consists of a weak acid and its salt with a strong base, and basic buffer consists of a weak base and its salt, strong acid. Example, ammonium hydroxide, which is a weak base, ammonium chloride, which is its salt. Buffer play important role in maintaining the pH of cells and cellular organelles to carry out their biological process. The important biological buffer in the human body are phosphate buffer and bicarbonate buffer. The phosphate buffer is an intracellular buffer in the cells which is maximally effect at pH 6.86 and has buffering range 5.9 to 7.9. The bicarbonate buffer is a physiological buffer system in human blood which plays crucial role in maintaining blood pH 7.4. The what is monosaccharide? Carbohydrates are divided into monosaccharide, three types carbohydrates, three types of there are three types of carbohydrates: monosaccharide, disaccharide, and polysaccharide. In monosaccharide रे कोड़ा रहेला aldoses. What are the aldoses? Glucose, ribose, galactose, and ketose रे only fructose. Ketose re only fructose there is a CO carbonyl group. Disaccharide it comes sucrose, maltose, lactose, polysaccharide, starch, glycogen and cellulose. Monosaccharide exist open chain structure. You can see monosaccharide. This is glucose structure. This is the alpha D glucose. This is beta D glucose. Alpha D glucose OH you can see here the carbon number 1 the OH is here and beta D glucose just opposite OH is this side, and this is fructose. This is the difference between the glucose and fructose. In the glucose there is a aldehyde group and in the fructose there is a carbonyl group. This is the alpha D fructose. This is the beta D fructose, just opposite. This is CH2OH OH, this is OH CH2OH. And derivative of monosaccharides undergo a number of reactions to form the derivative of monosaccharide. These react with the alcohol to form the glycosaccharides, and this is the glycoside. What are glycosides? Some monosaccharide aldose ketose contain both alcoholic group and aldehyde or keto group within the same molecule. The two group combine intramolecularly to give cyclic hemiacetal. You can see here this is okay. Uh, the two group combine the two monosaccharide join together, aldose ketose join together intramolecularly to give cyclic hemiacetal. 5 and 6 member cyclic hemiacetal and aldoses ketones are quite stable. D-glucose in the hemiacetal will be formed between the aldehyde C1 and alcoholic group at the C5. The carbonyl carbon D glucose, which has no chirality, becomes asymmetric following glycosylation and results diastereomers are formed. This is the open chain. This is the open chain aldehyde form of the D glucose, and this is the cyclic hemiacetal 2 stereoisomers. Further, the cyclic hemiacetal or hemiketal formed by monosaccharide can react with an alcohol to form the acetal or ketal. The acetal of a sugar is called glycoside, and bond between the carbon and alkoxy oxygen is called the glycosidic linkage. What are the glycosidic linkage? This plus this is the OH, and this linkage, if it is these two combined, C2H5 OH will go and oxygen will link with the two molecules. This is called glycosidic linkage. The glycoside of glucose is a glycoside. Glycoside of galactose is a galactoside, etc. If the pyranose or furanose name is used, the acetal is called pyranoside or furanoside. D-galactose as well as the D-glucose exist in nature as there are two amino derivatives and commonly referred as amino sugars. Amino sugars are formed when one of the hydroxyl group OH, but OH of the sugar is replaced by the amino group NH2 group. It is called beta D glucosamine, and by reaction with the galactose to form beta D glucosamine along with the acetylated derivatives, it is formed beta D N-acetyl glucosamine. Deoxy sugar, when one or more deoxy groups of monosaccharide are replaced by hydrogen, it is called a deoxy sugar. When one or more hydroxy group of the monosaccharide replaced by hydrogen, it is called deoxy sugar. 2-deoxyribose is one of such derivative in which the hydroxy C2 replaced by the hydrogen. Fucose is deoxy derivative of glucose in which CH2OH becomes CH3. And disaccharide. Disaccharide is most commonly surrounding oligosaccharides in nature. When two monosaccharide molecules join each other, it is called disaccharide. Disaccharide is formed when two monosaccharides get connected by a glycosidic bond. It is called. This is the glycosidic bond. This is maltose. You can see alpha D glucopyranose, alpha D glucopyranose, two alpha D glucopyranose molecule join together and water molecule goes out, and this oxygen, it is called glycosidic linkage. The two molecules attach with the by the linker, oxygen linker. This is maltose. This is called glycosidic linkage. And the alpha D glucose with the molecule of beta D glucose give a molecule of the maltose, a disaccharide. In this process, the alpha hydroxyl group of the alpha D glucose residue react with the C4 hydroxyl of the second residue with the elimination of a water molecule give the disaccharide. The linkage formed between the two monosaccharide, this linkage formed between the two monosaccharide is called glycosidic linkage and represented by 1,4. This is, you can see, this is carbon number 1, this is carbon number 4. This is called 1,4 linkage, 1,4 glycosidic linkage. Then polysaccharide. Polysaccharide, the name polysaccharide are large, high molecular weight molecule formed by condensation of the large number of molecules of the monosaccharide joined together by glycosidic bond. Large number of the monosaccharide joined together, it form the polysaccharides. Polysaccharides are employed as part of the structural tissue known as the structural polysaccharide, or may be used for storing energies known as storage polysaccharides. Then what is starch? These are the polysaccharides. The starch is the carbohydrate reserve form in the plants, which is a glucose polymer. Starch is a glucose polymer. In case of the shortage of the glucose in the cell, starch is hydrolyzed within the cell to release the glucose. Dietary starch is present in the potato, rice, wheat, corn, and many other plant sources. Starch is composed of two constituents, namely amylose. A polysaccharide with a linear chain is made up of D-glucose unit joined by alpha 1,4 glycosidic linkage and amylopectin. Cellulose, it is mainly responsible for the structure of the cell wall of the plants and consists of long chains made up of D-glucose unit joined beta 1,4 glycosidic bond. These chains are closely attached to one another by hydrogen or van der Waals bonds. And glycoconjugates. Glycoconjugates, oligosaccharides, and polysaccharide also exist in the covalent linkage with the lipids and proteins to form complex known as the glycoconjugates. Glycoconjugates believed to be more dynamic and information rich than the protein and nucleic acid. Glycoconjugates are divided in the following broad classes: glycoprotein, glycolipids, glycosaminoglycans, proteoglycans. Glycoproteins. Many proteins in the cells are covalently associated with the carbohydrate molecule, resulting in a type of biomolecule known as the glycoprotein. And what are the glycolipids? Glycolipids are the membrane lipids found in animals and plants in which oligosaccharides are attached. Structure of the galactocerebroside, which contains the has a characteristic 24 carbon fatty acids at constituent parts. Sugar molecule in the cerebroside can be sulfated in some cases. These sulfated glycolipids are known as the sulfatides. Some highly complex glycosphingolipids are derived from glycosyl ceramide and are called the gangliosides, which are present in the nerve tissues. And blood group proteoglycans, proteo and biology. So, what are the biological function of the carbohydrate? Carbohydrate, all form of carbohydrate are converted into glucose in the body, which then form a fuel to be burned in the liberated energy. Carbohydrate provided about 17 kilojoule of energy per gram. Glucose is converted into glycogen and stored in liver and muscles, where it acts as a reserve source of energy. Carbohydrates contains proteins and lipids are vital constituents of all cell membranes, mitochondria, brain and nervous tissue. Certain carbohydrates such as the galactose, amino sugar are component of blood group proteins such as the AB and O blood group proteins as proteins. So what are lipids? Lipids are a group of natural occurring organic substances that are soluble in organic solvents such as chloroform, benzene, ether, and highly hydrophobic, thus water insoluble. Most of the lipids are derivatives of fatty acids. Lipids are derivatives of fatty acid which are insoluble. Hydrophobic means insoluble. Lipids can be classified on the basis of their structure and function. Simple lipids are esters of fatty acids with alcohol such as glycerol, oil and fats are common example of this class of lipids. Fats are generally esters of the fatty acids with the glycerol. Complex lipids are also esters of the fatty acids with alcohol. Fatty acids are generally named after the parent hydrocarbon substituted for the last in the name of the hydrocarbon. These are the fatty acids are saturated fatty acid and unsaturated fatty acids. The unsaturated fatty acid with the double bond named as the enoic acid, octadecanoic acids, commonly known as the oleic acids. These are the unsaturated fatty acids. And fatty acid containing 8 carbon atoms are called the octanoic acid. Unsaturated fatty acids containing one double bond is called the monounsaturated fatty acids. The fatty acids having two or more double bonds is known as the polyunsaturated fatty acids. And what are the lipoproteins? Lipoproteins are the lipids being insoluble in water, cannot be transported in the free form in the circulating blood plasma. Hence, these are transported in association with the proteins. Such protein complexes with the lipids are called the lipoproteins. Lipoproteins are water mixable and consist of mainly of the tri, mm, tria, glycerols. Lipoprotein, lipids being insoluble in water cannot be transported in the free from in circulating blood plasma. Hence, these are transported in associated with proteins. Such protein complexes with lipids are called the lipoproteins. Lipoproteins are water mixable and consist mainly of the triacyl glycerols, phospholipids, cholesterol, and one or more protein compounds is called the apolipoproteins. Proteins, these are the derived lipids. The derived lipids are also called non-saponifiable lipids. These are the steroids. One of the common known steroid is cholesterol, which is widely distributed in body cells and is a major constituent of the plasma membrane and plasma lipoprotein. Terpenes are another large group of lipids which are made up of repeating isoprenoids units are the precursor of the steroids. Terpene molecules are derived by the condensation of the isoprene unit, which give rise to some important compounds are carotenoid. And terpene molecule derived by the condensation of the isoprene units, which give rise to such important compounds, is called carotenoid, from which vitamin A is derived, and cholesterol, from which several steroid hormone, bile acid, and vitamin D are derived. The structure is being indicated the isoprene unit. There are many isoprene units joined to form the vitamin A, to form the terpene. Vitamin A is also derived and cholesterol, from which the several steroid hormone, bile acid, vitamin D derived. What are bile acids? Bile acids are yellowish, brown or green fluid produced in the liver, stored in the gallbladder. Bile salts act like soaps and other emulsifiers. These contains both polar and non-polar regions, helping to break fats in foods in smaller pieces so that the food is hydrolyzed more easily. The structure of a bile salt is shown. So, structure of the bile salt. Okay, steroid hormone. Then hormones are chemical released by cell or glands in one part of the body and send out messages that affect the cell in other parts of the body. Many hormones are steroids. The steroid hormones can be divided into five classes: glycocorticoids, mineral corticoids, androgens, estrogens, and progestins. These are the structure. Yes, yes. These are the structure of the steroid hormones. These are the three steroid hormones are here. This is also another steroid hormone. estradiol, estrone. These are the steroid estrogens. Steroid and estrogens are female sex hormones. These are the estrogens. These are the chemical structure of the estrogens. These are the female sex hormones known as the estrogens. These are secreted by the ovary and are responsible for development of the female secondary sex characteristics. These also regulate the menstrual cycle. Progesterone is the hormone that prepare the lining of the uterus and implantation of the fertilized ovum and is essential for maintenance of the pregnancy. During pregnancy, it also prevent ovulation. The structure of the estrogens are given below. Estradiol, estrogen, progesterone. These are the three hormone, female hormone. This is another testosterone. Testosterone is male hormone. This is the male hormone produced in the test. It regulates the production of the sperm and aids in the development of the male secondary sex characteristics. Then eicosanoids. The polyunsaturated fatty acids. This is a fatty acid. Polyunsaturated fatty acids are essential for animal and human nutrition because they are the precursor to a class of compounds collectively known as the eicosanoids. Eicosanoids comprise of the prostaglandins. These are derived from the arachidonic acid, which contains four double bonds. There are one, two, three, four. Four double bonds are there. The structure of these three eicosanoids are depicted in this figure. And lipid aggregate. How the lipid aggregate? Lipid has the monolayer, and monolayers formed when the two lipid molecules are added to aqueous solution. The single layer formed at water air interface exposes polar head towards water and hydrophobic tails towards the air. Micelles are the spherical structure that contain anywhere from a few dozen to a few thousands amphipathic molecules. These molecules are arranged with their hydrophilic polar head group at the surface, hydrophobic and hydrophilic aggregation of the liquid, micelles, bilayer, and liposomes. Bilayer is another type of lipid aggregate in water in which two lipid monolayers are arranged back to back to form a two-dimensional sheet. This is the hydrophobic. This is the hydrophobic part, and this upper part of these lipids are the hydrophilic part. The hydrophobic person is in the hydrophobic person, the monolayer interact with each other. The hydrophilic head groups interact with the water. These head groups are called the hydrophilic part. That means these are soluble in water. The head group interact with the water as a surface and bilayer because the hydrophobic region as its edges are transiently in contact with water. The bilayer sheet is relatively unstable and spontaneously forms a third type of aggregate. It folds back on itself to form a hollow sphere, a vesically or liposome. Liposome are the closed self-sealing solvent-filled vesicle bounded by a single bilayer. Fluid mosaic model. What are the biological function of the lipids? Biological function of lipids. Lipids are important dietary constituents. On oxidation, they provide about 38 kilojoule of energy per gram. Fat is a poor conductor of heat, so a thick layer of the subcutaneous fat helps to prevent loss of heat. Lipids serve as the packing and support material to the abdominal organs like the kidney. Fat represents the most concentrated form of stored energy, and polysaturated fatty acids have a cholesterol lowering effect and are the dietary means of the preventing coronary heart diseases. Cholesterol is the precursor molecule for synthesis of the bile acid, steroid hormone, and vitamin D. So, from this unit, we learn that the lipid are a group of naturally occurring organic substance usually formed by combination of the fatty acids, glycerols, and soluble in the organic solvent. such as ether, chloroform, etc. Lipid can be classified on the basis of their structure into simple, complex, and derived type. And cellular organelles such as mitochondria, nucleus, lysosome, chloroplast are bound by membranes. The biological roles of lipids include prevention of loss of heat, packing, and support material. Then the biological role of lipids include prevention of loss of heat, packing, and support material. Most concentrated form of stored energy, cholesterol, lowering the effect, thus a dietary means of preventing coronary heart diseases. So this unit covers your oligosaccharide, monosaccharide, lipids, and cellular organelles. Then your unit 3, it contains proteins and nucleic acids. What are the amino acids? Amino acids which have their amino group attached this alpha carbon. This is known as, this is a amino acid. This is the alpha carbon. This is the carboxyl group is here. Acid group is here. The carbon atom attached to this acidic carbon is known as the alpha carbon. This is alpha carbon. This is side chain. This is the alpha amino acid. Those amino acid which have their amino group attached to this alpha carbon known as the amino group. Here you can see this amino group attached to the alpha carbon, so it is called alpha amino acid. The alpha carbon also carries a hydrogen atom. The fourth group on the carbon is known as the side chain and is denoted by R. This is the R group. The simplest alpha amino acid is glycine. The simplest amino acid is glycine. Glycine has a hydrogen atom as the side chain. Alpha amino acid, this side chain consists of long carbon chain and more complex chemical groups. All the alpha amino acid differ from each other in the nature of their side chains only. Thus, the general structure of an alpha amino acid can be represented as given. This is the alpha amino acids, alpha amino group. The structure of glycine, this is the structure of glycine. This is the structure of glycine. Glycine is represented by GLY and not as GLY. And then classification of the amino acid based on the nature of the R. There are different type of aliphatic amino acid, hydroxy amino acid, sulfur. If it is sulfur, if the R group is sulfur group, this is called sulfur amino acid. If the, depending on the what type of R group is attached to that group, aliphatic amino acid have aliphatic chains, R group, aliphatic chains, hydroxy. If it is R group is OH, it is called hydroxy amino acid. If the R group is a sulfur atom, it is called sulfur amino acid. If the R group is acid, it is called acidic amino acid. If basic amino acid, if the basic amino acid such as additional NH2 group is present, this is the additional NH2, it is basic amino acid. If the R group is a phenyl group, this is called aromatic amino acid. If it is the amino group is different from other, there is an imino group, it is called imino amino acid. Classification of amino acid based on the polarity of the R group. It is the non-polar R group. If the non-polar group has generally a non-polar aliphatic or non-polar aromatic chain, polar uncharged R group, the uncharged polar group have side chains contain the hydroxyl, amide, and sulfhydryl group. The charged polar group can be acidic or basic. Essential or non-essential acid. Our body can synthesize only 10 of the amino acid. These are called, body can synthesize 10 amino acid. These are called non-essential amino acid. And known as in the rest are synthesized in the human but are synthesized in either plant or bacteria. Essential amino acids are leucine, isoleucine, lysine, methionine, phenylalanine, tryptophan, threonine, valine, histidine. Proteins that contains all the essential amino acid are called the adequate proteins. Structure of L amino acid with three and other names. Non-polar R group, polar. You can see this table. These are the non-polar R group. Hydrogen, methyl. These are the non-polar R groups are there. There are polar group NH, COH. These are the polar groups. Polar. These are also polar. Polar uncharged R group. Polar negative charge group. Polar positive charge group. Characteristics of what are the characteristics of the amino acids? You can see this is a amino acid. This is dipolar means NH3 plus CO minus two dipoles are there, plus minus group is there, so dipolar amino acids. This is called Zwitter ionic acids. Uh, mm, An amino acid is usually represented in its own ionized form to show the presence of both amino and carboxylic group. In amino acid, both the amine group is there and carboxylic group is there. However, these are represented in the ionized form. You can see it is the ionized form which predominate under the physiological condition of the pH around 7. The understanding of the ionized structure of amino acid help us in understanding some of the properties of the protein. You can see here. This is the Zwitter ion. This is the anion. This is the anion. It takes when it reacts with the H plus. This is NH2 is converted in NH3 plus. This is the Zwitter ion. This is the anion, and this is the cation. When it is NH3 plus, it is NH3 plus. So this is the at high pH, this is the anion, this is the cation, and this is the Zwitter ion. Plus minus both the charges are here. So this is called Zwitter ion. What is isoelectric point? Isoelectric point. And the Zwitter ion have one positive Zwitter ion. What is called Zwitter ion? Zwitter ion has one positive charge and one negative charge. Negative charge are called the electrically neutral. Therefore, these dipolar ions do not migrate to an electric field. The pH at which the amino acid carry the net charge is called isoelectric point. For example, the isoelectric point of glycine is pH is equal to 6.0, which means that at this pH, glycine is in the Zwitter ion. The net charge is 0. Here the positive and negative pH around neutrally. This Zwitter ion, the net charge is 0. This is charge is -1, this is charge is +1, and this net charge is 0. This is called the isoelectric point. We can observe from this reaction H plus OH minus to a Zwitter ion results in the net positive negative charge on the molecule. Thus, depending on the pH of the aqueous solution, amino acid can exist in different ionic form. This property of amino acid to behave as weak acid or weak base is employed effectively in nature. These molecules act as buffers in body fluids. Thus, one of the most important functions served as proteins in the blood is to maintain the pH of blood in narrow range 7.35 to 7.45. The amino acid earlier separated from a mixture of amino acid are obtained by hydrolysis of plant and the animal protein. Extracted protein is then purified by dialysis and should not contain any ionic impurities. The separation of amino acid based on the migration of the amino acid in an electric field. At the neutral pH, the acidic amino acid will have net negative charge and will migrate towards the dipole. Positive pole while the basic amino acid will migrate towards the cathode, that is negative pole. Electrically neutral amino acid will not migrate unless the pH is made acidic or basic. Then peptide. What are the peptides? Peptides, two or more acids react together to form peptide. Two or more amino acid react. When two or more amino acid react with each other, it form a peptide by elimination of the water. Water will be eliminated when two amino acid reacted. The water will be eliminated and peptide bond is formed. The amino acid that could combine together can be same or different. We may call it as a condensation reaction. And the amide linkage, amino acid, alpha amino acid of other amino acid with the formation of the amide linkage. There will be a formation of the amide linkage. You can see here is a amino acid, NH2 group and acid group is there. Here is another amino acids. So this is form the OHS. This is water molecule can go away, water molecule eliminated. So CONS bond is formed. Another new bond is formed, CONS. This is amide linkage. This is the peptide. This is called peptide. This is residue of the one amino acid. This is residue of another second amino acid, and the middle is the amide linkage. This is called peptide bond. Separation of the amino acid, it is called paper electrophoresis. And you can see here another, this is one amino acid glycine, this is another amino acid alanine. When glycine and alanine join together, a peptide bond is formed, CONS, amide bond is formed, CONS, and this is the peptide bond. CONH group is there, peptide, that is called peptide bond, CONH. Similarly, tripeptide results. You can see here two CONH is here, another CONH is here. This is dipeptide bond is formed. Tripeptide, similarly, tripeptide also can be formed. Tripeptide bond can be formed. When the three amino acids join together, tripeptide bond is formed. Here two amino acids join dipeptide. When two amino acids join, one peptide bond. When three, it is two peptide bond is formed. Three amino acids join together, two peptide bond is formed. Similarly, tripeptide result from the three amino acid and a tetrapeptide link together four amino acids. Generally, peptide with more than 10 amino acids known as the polypeptide. You know, it is more than 10 amino acid, it is called polypeptide. Structure of the peptide bond. Then biologically importance of the peptide. Biologically importance of the peptide. Biologically important peptides are oxytocin, insulin, and glutathione, and bradykinin. These are the important peptides. And classification of the protein. This type of proteins can be classified based on their solubility in different solvents such as water, salt solutions, and alcohols. Shape and structure. Fibrous protein. Fibrous Fibrous proteins are the keratin, nail claws, collagen, bone, teeth. Globular protein examples are insulin, albumin, and chemical composition, simple protein, conjugated proteins, derived proteins, and their biological function, structural proteins. Proteins have central role in the mechanical support and stabilization of the many structures. Enzyme, all the living organism, almost all the reactions are catalyzed by specific proteins is called enzymes. Regulatory proteins, many hormones are like insulin, glucagon, and thyroid stimulating hormone. Transport proteins, specific proteins transport many organic and inorganic small molecules. Storage proteins are proteins which helps to store organic and inorganic molecules. Protective or defense protein, antibodies or immunoglobulins are glycoproteins that recognize antigens expressed on the surface of viruses. Structural organization of the protein. There are primary structure of the protein. The primary structure actually it is assembled like a linear long chain. Primary structure is linear long chain. This is the primary structure of the protein. Linear long chain polymer of the amino acid is called polypeptide. The number and sequence amino acid present in N terminal to the C terminal linked linearly by peptide bond. The complete covalent structure polypeptide constitute the primary structure. Disulfide, this is you can see disulfide. This is disulfide bonds are extremely important in stabilizing higher order protein structure. This bond may concern two polypeptide chains through cysteine residue. Weak non-covalent interactions in protein folding. This is a disulfide bond. SS. Weak non-covalent interaction in the protein folding. The weak non-covalent interaction that contribute to the stability of the protein structure have been identified as electrostatic force, van der Waals force, hydrogen bond, and hydrophobic interaction. Then secondary structure of the protein. Secondary structure of the protein basically the specific geometric arrangement of the amino acid that results from amino linkage that are close to each other in the polypeptide chain or a protein. These are the alpha helix structure. The alpha helical structure results from a particular pattern of the rotation around the N alpha carbon and alpha carbon carbon bonds of the polypeptide chain, which means the polypeptide chain can turn back under itself in a spiral or alpha helix structure. Alpha helix is thus a tightly coiled structure that resembles a right-handed screw or a circular staircase. And beta plated seeds in this arrangement, the polypeptide chains are almost fully extended and hydrogen bonding occur between the closed zigzagging parallel chain. Tertiary structure of the protein. Tertiary structure of the proteins are twisted, folded, packed into compact, almost globular three-dimensional shape, which is known as the tertiary structure. In the tertiary structure, stretches the secondary structure such as alpha helix and beta sheets along with the randomly coiled regions. And the tertiary structure is stabilized by hydrogen bonding and hydrophobic interactions, which are principally responsible for specifically of the folding. However, other interaction van der Waals dispersion forces to make a significant contribution. And quaternary structure of the protein. Quaternary structure of the proteins involves association of the two or more individual protein units. Relationship between tertiary quaternary structure can be best illustrated by the myoglobin hemoglobin. Myoglobin function as oxygen storage protein in the muscles. Myoglobin oxygen storage protein in the muscles, whereas hemoglobin transports oxygen through the body. Myoglobin binds oxygen. Myoglobin binds oxygen when O2 content in the cell is high and releases it when the level of the cell fails. A non-protein molecule called heme, which an iron atom in its center, is the oxygen binding site of the myoglobin molecule. It is located in the non-coplanar cavity of the myoglobin molecule and bonded to a histidine molecule in the chain. Hemoglobin is present in the red blood and cells where it binds oxygen and then transport it to tissue throughout the body. The nucleic acids, the building blocks of the nucleic acid, nucleotides, nucleosides. There are adenine, guanine, thymine, uracil, cytosine. The There are certain main common constituents. These are the phosphate group, purine base, namely adenine, guanine, pyrimidine base, cytosine. However, major difference between RNA and DNA is the former contain the pentose sugar ribose, where DNA contain two riboxyribose as the pentose sugar. Mhm. These are the ribonucleosides or deoxyribonucleoside. The common ribonucleosides are adenine, guanine, cytosine, uridine, and deoxyribose nucleic acids and ribonucleic acids. Deoxyribonucleic acid DNA is the fundamental unit of heredity. DNA has the pronounced secondary structure consist of the two independent polydeoxyribose nucleotides trans coiled round common axis and associated together by hydrogen bonds. Ribonucleosides RNA are polymer of ribonucleotide having 5 to 3 phosphodiester linkages. RNA, in fact, are the middleman of the informational transfer, depending upon the different role they play in biosynthesis of protein and polypeptide. Ribosome RNA. These are most abundant RNA and present in the ribosome in the cell. Messenger RNA. Messenger RNA are much shorter than the ribosomal RNA and are active RNA in the polypeptide synthesis. Transfer RNA takes parts in the amino acid transport system in the cell that carries individual amino acids. And in the summary, in this unit, we learned the what are the amino acids, what type of amino acids, and there are two techniques used separation of the amino acids are paper chromatography, paper electrophoresis. Two or more amino acids react together to form peptide by the elimination of the water molecule. And polypeptide with molecular weight more than 5,000 are generally called protein. The function of properties of protein are related to their diversified structure. Proteins are classified on the basis of their solubility, shape, structure. Proteins are organized. Polypeptide molecular weight more than 5,000 are called proteins. Nucleic acids are large complex biomolecules. In this unit, you also learn the nucleic acids are large complex molecules. They are found in the nucleus of the living cells. They are responsible for the heredity. Nucleic acids are polynucleotides. DNA is the universal genetic material. Ribonucleic acids are the other nucleic acid consisting of the nitrogenous bases adenine, guanine, cytosine, uracil. And there are three kinds of RNA depending upon the role played by them in the polypeptide synthesis. So the unit 4 is the intermediary metabolism. So what is metabolic process? Metabolic process metabolism is the sum of the all enzyme catalyzed chemical reaction taking place in an organism. The word metabolism is derived from the Greek word metabol. What is the outcome of this metabolism means breakdown of the complex biomolecules obtained from the environment into simpler usable compounds, efficient extraction of chemical energy of the biomolecule into ATP and reducing equivalents for the biosynthesis and other cellular activities, synthesis of complex biomolecules from simple precursor in accordance with the changing needs of the organism, synthesis and storage of long and short-term energy reserves in conditions of excess. Metabolic breakdown of the carbohydrate, fat, protein shows that these may be considered to proceed in three stages as in the first stage, large nutrients are hydrolyzed to smaller molecules which are then building blocks. What is catabolism and anabolism? Catabolism involves breakdown of the complex biomolecules, so such as carbohydrate, lipid, protein. They break down to some smaller molecules. These are called catabolism. Protein into simple molecules such as carbon dioxide, ammonia, water. These reactions are energy generating and oxidative in nature. Some part of the energy released in utilized for the synthesis of ATP reduced energy carriers and these are the catabolism. Catabolic sequence are convergent in nature, and anabolism is divergent in the nature in the sense that diverse set of compounds, namely carbohydrate, fats, proteins, are synthesized from a common set of precursors. Design of metabolism reaction that occur in all the organisms are very large number. Unicellular organism is capable of catalyzing a few thousand reactions. Introduction to bioenergetics. The structure built from the complex organic molecule is possible by continuous flux of energy through the living system and transformation of various processes. These energy transformation in the living systems are governed by the laws of thermodynamics. The study of such transformation comes under the view of bioenergetics, which include the generation of the energy by breakdown of the nutrients, storage and utilization in performing various functions of the cells. This is the additivity and LG value coupling reactions. So, the biological energy carrier, hydrolysis of ATP. Adenosine triphosphate is a nucleotide, consists of purine base, adenine, a ribose sugar, and a triphosphate unit gives structure of the ATP. It gives hydrolysis product, namely adenosine diphosphate ADP, adenosine monophosphate AMP, and inorganic phosphate. A large amount of the free energy generated on the hydrolysis of ATP is given in the figure, in the page number 118. Role of ATP in the biochemical energy transfer. ATP continuously formed and consumed in living organism. It acts as energy coinage or energy currency of the living cells. An analogy of the role ATP in living cell is seen in exchange of currency when you visit a foreign country. A role of ATP as energy transducing agent and or vehicle suggested that it should be rapidly turned over, rapidly or rapidly broken down to ATP. When a glucose molecule when react with oxygen, it gives carbon dioxide and water. Glucose molecule. Before we go into the chemistry of ATP ADP interconversation, it is important to compare the free energy change involved in this process with that obtained from the oxidation of a common nutrient. When glucose is oxidized, it gives carbon dioxide and water. A large amount of free energy become available on complete oxidation of the glucose into carbon dioxide and water. For an efficient utilization of the energy for the synthesis of ATP, it is necessary that it should be liberated in the several smaller packets, each of which adequate to drive the synthesis of ATP. Structural basis role of the ATP. When the glucose molecule react, when you see del G0 value for the hydrolysis of ATP is much larger than the AMP. In fact, to be found, it is found that larger than the hydrolysis of all simple phosphate ester, glyco 1 phosphate, glucose 6 phosphate, etc. The reaction of hydrolysis of glycol, glycerol 1 phosphate, and corresponding value of del G0 is given below. Del G0 for it is minus 9.205 kilojoule per mole. ATP, ADP have been referred to as high energy phosphate compounds, and the phosphorus anhydride or pyrophosphate bond between the phosphate group and these compounds have often seen referred to as the high energy bond. High energy phosphate compound would participate in the metabolic process. Some compounds other than phosphate derivative also exhibits large del zero value for hydrolysis. What is universal electron carrier? Universal electron carrier cell obtain free energy in chemical form by catabolism of nutrient molecule and they use that energy to make ATP, ADP and the organic phosphate. ATP then donates some of its chemical energy to endogenic processes such as the synthesis of the metabolic intermediates and macromolecules from smaller precursor molecules, the transport of substance across the substance against concentration gradient and mechanical motion. Cellular oxidation of glucose to carbon dioxide require specifies electron carrier. Molecule that accept electron from electron donor and donate these to electron acceptor creating energy producing electron transport chain. Homeostatic control system and component. What is homeostatic component system? Homeostatic control system Homeostatic control system is an important part of the human body to maintain the physical chemical state as constant equilibrium and constantly monitoring the internal variable. Homeostatic is regulated by the four component of the homeostatic control mechanism. These are the stimulus, receptors, control center and effector and explained in the stimulus, sensor, control center, effector, component of the homeostatic control system. Stimulus, sensor, control center, and effectors. Maintaining water balance. The amount of fluid in the body is tightly controlled because imbalance can be devastating. In a normal individual, the maintenance of the water balance is achieved by adjusting both water intake and excretion. The water balance in human beings in cool environment. Regulation of the water balance. The input of water as well as the loss can be highly variable due to the individual habits and environmental factors. In spite of this, the total body water needs to be maintained constant to achieve the normal osmolality of the physiological function. Osmolality is a measure of the osmoles of solute per kilogram of solvent. In chemistry, osmol is the unit of measurement that defines the number of moles of a chemical compound that contribute to a solution's osmotic pressure. Kidney. The kidney are highly efficient conservator of body water, although kidney filter 180 liter of the blood per day. The urinary output is in the range of 1 to 2 liter per 24 hours. Before the urine leaves the kidney, variable amount of water and various solutes are reabsorbed by the renal tubules. Hormonal control of fluid balance. When water intake is insufficient or water loss excessive, the kidney compensate by conserving water and excreting more concentrated urine. The renal tubules released by the pituitary gland under the stimulus of the hypothalamus and this helps restore the blood volume. Cells in the kidney responds low blood pressure by releasing enzyme called the renin, which in turn cause the kidneys to reabsorb sodium through a series of events. Sodium reabsorption in turn accompanied by water retention, thus helping to restore the blood volume and the blood pressure. Renin activates a protein is called angiotensinogen in the blood to give the active form angiotensin. Being a powerful vasoconstrictor, angiotensin narrows the blood vessels. In summary, the three mechanisms effectively restore the homeostasis by responding to low blood volume and highly concentrated blood. The kidney responds to reduce the blood flow by releasing enzyme renin. Then renin initiates the activation of the protein angiotensinogen. Then kidney retains sodium and water, thus increasing the blood volume. So in this unit, we learn in the summary In the summary, in this chapter, unit 4, we learn the metabolism. And metabolism is the sum of all enzyme catalyzed chemical reaction taking place in the organism. All the metabolic reactions, particularly either catabolism, anabolism, or both catabolism refer reactions involved in the breakdown of the complex biomolecules such as carbohydrate, lipid, protein into simple molecules, carbon dioxide, amine, and water. Bioenergetics is the generation of the energy by breaking down of nutrients, storage and utilization in mm, performing various function of the cells. Living organisms differ markedly in their appearance. The ATP synthesis about 50 to 60 kilojoule per mole. Cell converts glucose to carbon dioxide not in a single high energy releasing reaction but rather in a series of controlled reaction. All the cells in our body are surrounded by liquid called the tissue fluid, which has exactly the right condition in which cell can survive and work. The homeostatic control system is a signal directed mechanism which works in a feedback loop and can occur at organ, cellular, or molecular level. This is done by negative and positive feedback mechanism. And the amount of fluid in the body is tightly controlled because imbalance can be life threatening. In normal individual, the maintenance of water balance is achieved by adjusting both water intake and excretion as needed. There are a number of ways of water input and output. The regulation of water balance is affected by digestive system, kidney, hormonal control, and thirst. So this is the last unit where you learn what is metabolism, what is bioenergetic, and what is homeostatic control system. So all this system. So the questions are there, and you can do yourself the solve the questions yourself by reading all these units. So this is over now. Okay. Hello. Hello. Hello.
Yes, ma'am.
Yes, ma'am. Okay. So now it is over.
Thank you, ma'am.